The cortex, we can divide it based on its function. But there are landmarks of the cortex, these grooves and this folding. In most people, they are relatively conserved. So based on the major grooves on the cortical surface, we can divide the cortex into different parts. This is just a landmark. Based on this landmark, we can say we can identify various regions of the cortex responsible for different functions. For example, if you have a stroke that hurts, that causes some lesions in the left side of the prefrontal cortex, you lost your ability to speak. Right? The language area is right there. If you have a lesion right here, the motor cortex region, you lost your ability to control your motion. So, and also the other area for sensory processing, every part of the cortex appear to be devoted to a particular function. So there's a cortical localization of function. This is one of the major things of that people have understood over the last century. Now, if you look at detail, you cut a cross-section of the motor cortex, you'll find that if you get a piece of cortex here and look at the function within this cortex, you'll find that the cortex even subdivided into more subregions. For motor cortex, a different region of the motor cortex devoted to different parts of the body. The hands and fingers and arm and legs are controlled by different regions of the cortex. Cells in here controls particular muscular action. So if you stimulate this region, your leg would move. If you stimulate here, your hands will move, your finger will move. So there's a map of the peripheral surface to your cortex, even more defined subdivision. Now this cortical map, interestingly, which was first discovered or finally characterized by a Canadian neurosurgeon, Penfield. When he was doing neurosurgery, he wants to figure out which region he can perform the surgery to cure the epilepsy. So you have to do stimulation of every region to find out which is the region that causes epileptic discharge. And doing this, he found out there's a map of the control different part of the motor system. And also there's a map control different part of the somatosensory system. This region a little bit back on your brain, there's a somatosensory cortex. This region controlling the face, the sensation from the face, from lips, from hands, and from different parts of the body. So this map is highly distorted. If we take the same proportion as the area of the cortex devoted to a particular function, you would make a figure, and this is what you wrote with a homunculus, a small man within the head controlling our motor system and sensory system, a highly developed hands. Now that's what will make us human. Because our hands is much better, much more flexible than a monkey or chimpanzee. That makes us tool-builded. So the hands and face for facial expression is so important for social communication. That's how specialized in human. Evolution has produced a genetic mutation that allows these particular functions to develop. Now, gene is responsible for this, but being able to use it, and this region gets even more enlarged. This topography are similar to among people, but they are also highly plastic. In fact, one can do an experiment. You train a monkey to use 3 fingers instead of 5, and the area occupied by these 3 fingers, this 2, 3, 4th finger, will be enlarged after a few months of practice. Right? Now you can imagine a piano player, their cortical area devoted for their fingers are much larger than ours because they've been used, and more cells are devoted to it to control the actions of the fingers, and that area gets enlarged. And in fact, in human, there's an interesting experiment using brain imaging method. You look at the area occupied by string players in an orchestra. The string players, their cortical area, motor cortical area, controlling their left-hand fingers, which they use a lot for string playing. The left-hand fingers are much more, the area is much bigger than the non-string player as a control. Right? Those who started playing earlier at a young age, their area is larger than those started playing later. So this very interesting non-invasive studies on the area devoted to left-hand finger tell us two things. The first thing is that there's the earlier you start using this area, you have larger area devoted to it. Right? So there's a age-dependent changes. The second thing tells us that even if an adult, you are already mature in adolescent, even in adult, you still have a certain plasticity. Use the brain in the mature brain still change the size of the cortex devoted to that particular function. Right? So there are two things: early developmental plasticity and then later adult plasticity. The adult plasticity, we know it's all, everybody knows, the younger kids learn better. But the old person, adult still learns. So there's graded change in the plasticity. When the mechanism for this plasticity may be very similar. So the one interesting thing that I was going to come back to the issue, take-home message is that even in adult plasticity, the early developmental plasticity may be buried there. One can reveal the early developmental plasticity by particular experimental protocol. Right? So you can reawaken the developmental plasticity, and that's very important for therapy. Right? When you have an injury, how you regain the function. Now you don't have the early developmental plasticity to form a network anymore, but you, by proper approach, if you know how to do intervention, to rehabilitation, you could reawaken this plasticity, early plasticity, and regain the function. Right? This is a very clinically important aspect.